So hello and welcome to another episode of Talking with Experts. I'm often asked for my opinion regarding feeding DFM to honeybees. So today we're delving into Strong Microbials, SuperDFM, and my guest is Dr. Vera Strogolova, a scientist and co-founder of Strong Microbials. I'm Frederick Dunn, and this is The Way to Be. Here's Vera in Milwaukee, Wisconsin.
Hi, my name is Vera Strogolova, and I am a scientist, a beekeeper, and a co-founder of Strong Microbials. Strong Microbials is a company that we started with my husband 10 years ago when I was in graduate school, actually. And we started it because we had a lot of experience working with probiotics for animals. And it was just really a great, interesting topic for us. We liked knowing where your food comes from, and we liked doing applied science a lot. And what I do now at the company is I'm a scientist, so I do oversee a lot of research trials, but also day-to-day operations and quality control because we grow bacteria. And so what we do is we have big brewing tanks, and these brewing tanks are fermentation tanks. And we are working on growing different strains of bacteria and then seeing what happens with them when we go to an apiary and we can actually give them to the bees as probiotics.
So, okay, that's really interesting. And I'm going to jump into a lot of different categories of what you've worked on and studied in the past. And I want to thank you for being here, because a lot of people want to know, the viewers are likely to be backyard beekeepers, and they're located kind of all over the world. And I'm sure most of them have heard of SuperDFM, so Direct Fed Microbials, or is it Direct Fed? Is that correct?
Yes, yes, that's correct.
Okay. So
yeah, that name comes from our background. You can just say probiotics, but when we were working on these projects, we called these probiotics direct fed microbials because that was the term that was used in the industry. So at first we worked with poultry industry and calves and dairy industry. So that was just a term that was accepted, and that was just something that everybody knew at the time. But then later we realized that DFM doesn't mean anything to a beekeeper or pretty much anybody other than outsiders.
So is there a big distinction between probiotics and DFM?
They actually are the same thing.
They're the same. Okay, that's good to know. And another part of this,
it's like interchangeable terms that you can call it tomato or tomato.
Tomato and tomato. Okay, that's good to know. And I also like that your background does include poultry, swine, and cattle. So you've been looking into providing healthy gut environments for a variety of different animals, and have now focused on bees, or are you equally focused on any other animal species?
Right. So, right, Fred. So we started doing this work with all of the different animal species, with poultry and with swine and dairy and cattle. That was an area that was using probiotics or DFMs for probably the last 30 years. And when we started the company, we didn't know anything about honeybees, and beekeepers didn't know anything about probiotics. It was kind of a new connection, a thing that made us really, really excited. Mm-hmm. And so I think that there is a lot of literature about probiotics or DFMs in animals and in poultry, and some of the work that happens there is more established. Whereas when we started working with beekeepers, it was just so new. Nobody really knew what to do about it. Mm-hmm. So I still spend most of my time thinking about beekeepers, really, because it's just so new, and so few people have been in this field. It's growing now. I'm really glad to see that there are more people who are interested in this question. But I think that was just something that was really exciting and continues to be exciting even now.
Mm-hmm. That's interesting. Did you take a personal interest in bees yourself? Like, are you a beekeeper? And is it a co-contributing thing? So what you're doing for your profession also blends with something that you're doing in raising bees?
Yeah, I think it's a kind of interest that is, of course, self-motivated. I mean, I'm interested in studying bees,
Okay.
and we have 20 some hives on the roof of the building, and everybody is involved. Everybody who works, we have a staff of about 10 people, and everybody loves to come up and help with the bees or honey extraction.
Mhm.
So I'm not a solitary beekeeper, and I don't do much heavy lifting, but yeah, we have to have this hands-on experience because we started with bees in 2014 without much experience and without knowing very much about bees. Our family were not beekeeping families.
Mhm.
And I think that from the scientific and from personal perspective, they're really interesting creatures. So beekeeping is enjoyable, but also good for work.
Yeah, and you said that you have a staff for this rooftop apiary. So you have interns that come and work with you as well?
Yeah, we had interns before COVID.
Oh, okay.
Hopefully we'll get back into that, but we just have, so we're actually manufacturing the products, the products that we sell. So these probiotics are manufactured here in Milwaukee, Wisconsin. And so everybody who works with us in the office and in the warehouse also loves to come up and help with the bees as well.
Look at the bees and maybe hang out with them and just see what they're doing.
Mm-hmm.
That's one of the
Sometimes
best.
do alcohol washes.
So we're killing the bees. Okay, well, it is necessary to assess the bees as well. Now let's backtrack just a little bit and assume that the people watching don't understand what the microbials are. So we're talking about living things, right?
Yes.
So if you could
microorganisms are everywhere, and it's just very small. So if you're thinking about bugs and insects being much smaller than animals, but still being very much alive and having very complex social organized life and having such a big impact on all levels of the ecosystem. And the same thing is true about microbes. So microbes are much smaller, so we cannot really see them without a microscope. But just like molds and mushrooms and bacteria and viruses, we know that there is a lot of life and a lot of things that are important for the health of the ecosystem and the health of animals and insects. And nothing is really sterile. So everything is alive in nature. So all of the surfaces, each blade of grass actually has a lot of bacteria and fungi and microorganisms on it. And for the most part, it's a pretty balanced ecosystem. So if we're looking at a honeybee gut, there is billions of bacteria in there, but it could be a very healthy situation in most cases. Even though some bacteria that are present there are maybe opportunistic or pathogenic, but usually you have more than one type of bacteria, and that balance usually keeps disease at bay. Mm-hmm. So that's what microorganisms are. And like I said, they're just kind of everywhere, right? So it's a part of nature. And if we use antibacterial hand soap, we kill some bacteria, but not much time goes by without bacteria coming back.
Mm-hmm. So with that in mind, how did you identify what the honeybees would need in their microbiome, or however you would describe it? In other words, how did you identify a deficiency? And then how did you select which bacteria or which what would you even call them? Which bugs would help the bees? So I guess.
Yes. Yeah, so much of that work is still ongoing. So we still are learning a lot about human microbiome and about animal microbiome and about the bee microbiome. So all of the community of different bacteria. And I think, right, like bugs, or I think about bacteria as seeds. So you can have different prairie mixes for your yard or for your land, right? So it's just different seeds. And we select certain ones that we know are good for the bees. We select others that are good for the butterflies. And so when we were selecting different bacteria for the bees, what we did is we looked a lot through literature search to identify the bacteria that were fed to honeybees in a scientific study. And we pulled out a couple of them which are not specific to honeybees. But we know that just like the seed mixes, those are kind of good bacteria. Lactic acid bacteria are usually thought, frequently are found as probiotic bacteria for many different species. And so we just looked for the bacteria because we didn't know so much about honeybees. So we looked for the existing literature about which bacteria could be given to honeybees, and the honeybees will eat them. And, we looked for studies that established some kind of benefit. For example, we looked for bacteria that could inhibit some of the honeybee pathogens. So for example, the fungus that causes chalk brood, it's very easy to study it. And we know that some of the bacteria are already identified that could kill or inhibit that fungus. So those are the candidates that we went to.
So then is that the initial malady associated with honeybees that you decided that you could work with once you could isolate the chalk brood bacteria and then understand how it would act on the probiotics that you're going to introduce?
We started by looking at what kind of benefits we could think of for honeybees. Another thing, like you said, identifying what was missing. There was a lot of study and research done already on the bacteria in honeybees. And then one interesting study was done on CCD, colony collapse disorder. So the scientists, before we got into bees, other scientists were looking at CCD colonies and comparing the microbiome or bacterial community of the CCD colonies and healthy colonies. And they were looking for like a pathogen like chalk brood, right? That would make them, the bees, that would cause CCD or make the bees sick. They didn't find any particular bacteria that they could pinpoint. But one of the things that came out that was interesting in that study is when they compared the healthy and the sick bees had much less lactic acid bacteria. And that's the class of bacteria which are often found as probiotic bacteria and are kind of like the prairie mix, right? They're usually good for many different purposes, and they help digestion in honeybees, but also in other insects and in other animals. So we kind of went towards that direction to knowing that maybe it was missing in honeybees, and this class of bacteria is usually a probiotic and could deliver benefits to the host.
So then once you introduce your microbes, how do you quantify the results in the bees? How do you do assessments to determine that it was beneficial or maybe didn't cause any change? Or how did you assess colonies after treatment?
Yeah, so it's not easy to study honeybees because there are so many factors, starting from which breed of honeybees you have and the geography, the location, beekeeping practices, which are so diverse. So one way that we study is we take one apiary and we work with the beekeeper to only give SuperDFM to half of the colonies. So when you do that, you have a control group, which doesn't get the DFM, but everything else about those colonies is the same. And one of the things that we look at is, so we look, for example, for things like colony size, how many frames of bees there are, and just tracking that over time.
Mm-hmm.
Another thing we looked at sampling the bees and doing lab analysis on them to find out their nosema counts, as well as their lactic acid bacteria counts. So lactic acid bacteria counts are the lactic acid bacteria, those probiotic bacteria. You would normally have a certain level of lactic acid bacteria in bees, which is considered healthy. And we can see if we are treating those bees with antibiotics, you would have less lactic acid bacteria. And when we feed them with probiotics, we could see that that was going up. So those kinds of studies, on one hand, you are looking at the health. So for example, the colony size, frames of bees, brood patterns. And on the other hand, you could look at pathogenic or parasites or pathogens, and you could look at disease, and you could look at lactic acid bacteria, which are the good bacteria.
So do you have beekeepers that are cooperating with you and doing studies in different locations around the country? And if you do, what's a ballpark of how many beekeepers are participating and how many hives are represented?
I think we worked with probably tens and hundreds of beekeepers over the 5-6 years. Some beekeepers are like our apiary, for example, is always a part of our study. Many beekeepers come year after year. Most beekeepers only look at one particular question for one time because it's very time consuming we usually ask to send the samples to us or to send the samples to another trustworthy source of information like Beanport Partnership. And so we have been doing that about every season.
So has the feedback been consistently good, or are there some neutral responses, or what are you hearing back, and what are the samples looking like that they're sending in?
Yeah, so usually, yes, we do have really good results, and we usually have very good feedback. And it's much easier to get positive words than actually to collect enough data, robust data, information that we could actually process and have good results in the lab. For the most part, everybody is pretty happy. And sometimes, and I think that usually you would see most difference when there is some challenge. So obviously, if the colonies are healthy and they are in pristine location, there isn't very much challenge, which is good.
Yeah, they may not need it.
And I think that with probiotics in other areas of agriculture, that's just kind of the rule of thumb that you have. So if you are using probiotics when you have some challenges. So if we're thinking about beekeeping industry, there is a lot of challenges, starting from just chemical exposure and pesticide exposure in the environment, because bees, unlike some of the other agricultural livestock, bees are different, right? They forage in a large area, and it's not necessarily the land that beekeeper can control. And also beekeepers and bees do pollination. And I think that also exposes them to not very favorable conditions.
Well, yeah, that's a big topic, of course, for commercial beekeepers that are pollinating, like right now in the almond groves in California. Now, I noticed that you have two different DFM lists here. One is SuperDFM-P801, and then you also list separately SuperDFM by itself. So is P801 specific for some agricultural exposure? Is there something that it does that the other one does not do?
Yeah, P801. So what we did is we originally formulated 7 bacteria in SuperDFM, and in a few years ago, we were looking at the different bacteria, Pediacoccus, P801, that was the bacteria that we added. And there was really interesting results, and two other groups, I think, were studying the same bacteria too, and showed that it really helps to protect bees from pesticides. And we were asked a lot, about pesticides. That was one of the biggest challenges that beekeepers were feeling and experiencing. I mean, they're bees, but that's when we decided that it would be good to add one more bacteria because we could see clearly that there were some benefits from it. And we did a trial that showed that it really helps to increase the weight of the hives and mortality. And that's what P801 is one more bacteria was added to SuperDFM to make it maybe perform even better in where there are pesticides.
Yeah, so I'm guessing then that the P801, the P stands for Pediacoccus?
Yeah.
Okay, and then how exactly does that deal with the pesticides? Does it serve as some kind of binder so that it doesn't impact the bees and instead goes through their digestive system and exits the bee? How does that work?
Yeah, that's one of the mechanisms. So I think that the study that actually delved deeper than our trials did into the Pediacoccus suggested that there are more than one mechanism in which Pediacoccus protects bees from pesticides. One could be that it stimulates detoxification systems of the honeybee. So honeybees actually have some of the enzymes which help them detoxify poisons or chemicals. And we all do. I mean, humans, we also have some ways to detoxify some of the poisons or toxins that we encounter. But bacteria also play a role because they can also have those enzymes too. And then indirectly too, so the Pediacoccus could also bind and absorb some of the toxins and pesticides, and that could be another mechanism. So we don't know right now which one of those mechanisms is important. Most likely it's a combination of those, and it will take more work to really kind of say like it's 50% the detoxification enzyme and 50% binding.
You're still doing trials with these formulas then, or these bacteria?
Yeah, so our research is more applied research. So we're looking at survival of the bees, for example, and more academic or more basic research would look at which particular molecules and which particular enzymes are responsible. And that's the challenge that we always face, right? On one hand, you want to do the lab study that would say like, this is exactly what happens and would be very mechanistic. But then you want to make sure that this kind of research works in the field. Right. And especially with bees, you know, the research that works with bee colonies is so different. You know, it's not a very clean lab setting. You can't put bees in a cage and say, you know, like I have my 20 bees and 200 bees. You know, you have to have hundreds of colonies to really get answers. And it doesn't help if it only works with one particular management style, because there are a lot of different management styles in beekeeping.
So who would you say, what kind of beekeeper is your biggest recipient of SuperDFM from your company? Are these backyard commercial people? Who's the most likely to want to use SuperDFM?
You know, I feel like we engage equally with small scale beekeepers, backyard beekeepers, and commercial beekeepers as well. But of course, commercial beekeepers have many more colonies. So that means that we probably have a lot more time and collect a lot more information from commercial beekeepers than from backyard beekeepers. So I would say it's both.
So when people are looking at this and, you know, somebody's sitting there, they've got 3 hives in their backyard and they get excited about it and think, I really want to improve my bees' digestive health and everything else. What kind of treatment cycles are we looking at? How frequently would they treat their bees and how would they deliver it to the bees?
Yes, so what we found is that it works very well to inspect and work with the bees every month And that's the frequency that seems to work very well for probiotics, for DFM as well, just feeding it once a month.
Okay.
On one hand, it's convenient for the beekeeper because obviously, some of the studies do it every day or every few days, but that's just not practical for anybody. And a brood cycle would be a couple of weeks. And we do know that when the bees are just coming out of their cell, they consume the hive materials around them, and that's how they get their bacteria in their gut for the first time. Because when they come out of the cell, they don't really have any bacteria in their gut yet, because they are just coming out, and they're very much empty vessels at that point after going through the brood transitions and pupation. So what happens is that you actually have nurse bees and just hive environment that gives the bacteria to the next generation of bees. And since in a month you have a couple of generations of new bees being born, it's good, I think, to repeat the application of these bacteria every couple of brood cycles, 2-3 brood cycles, and that works out to about once a month. That's the feedback that we had from some of the beekeepers who have more experience than we do as well. And like I said, it really matters what kind of conditions the bees are exposed to. So if there is less challenges and you're not really working with your bees for 2 months, I would say there's probably no harm in skipping a dose. But on the other hand, when we're working with commercial beekeepers who are doing more than one pollination a month sometimes, that's when it really becomes important to make sure that those bees are protected, and that's where DFM comes in about once a month.
And is it necessary, like you said, this coincides with when they're doing an inspection, for example, because they're opening the hive anyway, roughly once a month, you suggest. Is there another way to introduce it to the hive without opening?
Yes, some beekeepers actually really requested that they would be able to introduce it to the hive without opening. And we work with some beekeepers who use air compressors, and so we have air compressor attachment, and the air compressor attachment could deliver through the entrance, through the hive entrance. So that's called microcloud. It was an idea that a beekeeper that we worked with had, and we just kind of rigged it together, and it seems to work pretty good. It's like a little scoop that could slide inside of the hive.
I've looked at it on your website, and the reason I ask the questions is not because they haven't looked at it, so that people that are listening, because this is also a podcast, so we need to be descriptive. So, but yeah, it's a low pressure air nozzle, and then it looks like a piece of PVC piping material on the end, and you put the DFM in that, stick that in the entrance, give it a little puff, and you call that a microcloud?
Yes.
Okay.
Yeah, that's right. You described it very well, and I kind of neglected to give that good description.
Yeah, and I have an idea, by the way, for that, because when I was looking at it and I was thinking about it, have you considered an entrance reducer on the hive that actually, if the face of the hive were here and your entrance, here's your landing board, if you had a channel that went out, and if it had a reservoir in the channel where you put the DFM, all bees that are returning and going through that would then, of course, go through it and track it throughout the hive. Would that be a way to get the DFM spread throughout the hive as well, instead of the air puff?
That might work.
Yeah.
That might work. So it would be at the entrance.
Yeah, because
I know that there are similar setups like that too for other things that bees can transport. Yeah, that might work too.
Yeah. Okay. I'm just throwing my ideas in there.
Good idea. Yeah. Sometimes we work. So I have one. I usually work with Langstroth hives, right? So we get questions from beekeepers have different designs of their hives.
Mhm.
And we always say, send us the questions because we'll talk to some of our beekeeper mentors
Mhm.
and they will help us figure out how to answer this question. And yeah, so different ways to put it, to give it to the bees,
Mhm.
all might work, right? We just want to make sure that, I think that most importantly, the young bees and nurse bees, that's the ones that we want to.
Yeah, so you have to
keep
get it on those
it.
brood frames. Yeah.
That's why we put it next to the brood frames. But like you said, if you put it by the entrance, the bees will also share it because they do actually share lots of things throughout the hives, food and bacteria as well.
Yeah. Okay, so that's interesting. Another thing people may be wondering about is because these are alive. So what are the parameters where, like, what's a high temperature that might kill them, and is there a low temperature that might kill them?
Mm-hmm. Yeah, so we actually freeze and freeze dry lots of our ingredients, lots of our bacteria, so they're all dry. So low temperature is totally good. Low temperature actually preserves the bacteria very well for a long time.
Wow.
But you want to avoid temperature that is more than 120 degrees because that will kill the bacteria. More than temperature though, you want to avoid the humidity because we dry the bacteria so that they are like the dry seeds, so that they don't metabolize. Once they, for example, once the bees consume it, they are in contact with moisture. So that's when these bacteria wake up, and that's when they start to metabolize. So you want to minimize humidity, and that's why we have our products in mylar packaging, which prevents any humidity from getting in before you start using it.
Mm-hmm. So when they get this mylar package with their SuperDFM in it, what's the shelf life? How long can they store that and have it still be viable?
The shelf life of a package that is not opened is 5 years. That's kind of just the longest that we have tested it for. And once you open it, there is a Ziploc bag, Ziploc closure, a zipper on the top. Mm-hmm. And so you can seal it and you can store it without moisture, hopefully.
And so once it's been opened, how long is it good for?
It depends on how much moisture they're exposed to. So if it was opened and closed and ziplocked again, probably still good for many years.
Okay. Just because people always want to know, if I buy it, how long is it good for? So
yeah, absolutely.
Okay. So now, and so the bees, you say the bees eat it. So the workers also ingest this. I mean, how do they, or what encourages them to eat it?
Sucrose is the ingredient that is stabilizing the bacteria and also is tasty to the bees. So we have sucrose as one of the ingredients. And so, yeah, they eat it like they would eat a little bit of powdered sugar.
So is it powdered sugar?
Sucrose is sugar, yes.
No, but this is powdered sugar that they come in?
Powdered sugar is the carrier for the bacteria, yes.
Okay.
It's one of the ingredients.
Okay. That's good to know.
So what it does, it prevents, yes, it is very tasty and it is very palatable, but it also prevents moisture from, absorbs the moisture. So if there is any humidity, it keeps the bacteria stable until it gets opened.
So it acts as a desiccant.
Yeah.
That's interesting to know. And I have my notes. I have to go through them here really quick to make sure. How do they eat it? Best way to apply it. Oh, are there any current studies that are going to be published about the SuperDFM, or has already been published that we could share?
Yeah, all of the studies, all of the things that we've done with it are field trials. So I'm working on putting all of the information together, maybe, but I think that it will probably take a much larger scale study to get information that could be published, because usually you want to have more than just one experiment and more than just one trial
Right.
And it's very important to have standardized methods. So we would have to do more work ourselves, because relying on citizen science approach and the beekeepers who are doing these field trials is great, but it doesn't give us the data that we could easily, submit to a journal and get it published in a peer-reviewed journal.
Okay, so for anybody that's watching this in the future, when those results are published, they will show up, the links will show up in the video description, so people can look it up later. So if it's not down there right now, it's not available. So that's good.
Yeah, we put some of our field trials and some of our results on the website, but it's not the same as having.
And we will also link the website so people can see how it works and what's been done.
Absolutely.
And what is the dose? So when
one tablespoon.
1 tablespoon per
1 tablespoon per hive, per colony.
For the entire, okay. So no matter how many brood boxes, it's 1 tablespoon.
Yeah.
Okay. So what else can we talk about? Is there anything else that you want people to know about the company, or where you're headed as far as something else coming out, or what should they know in general about your company, Strong Microbials?
Yeah, so I think that one thing that I wanted to say is that using probiotic or microbial products is really interesting and appealing to us, because it's an alternative to using chemicals, antibiotics, and pesticides. So we try to work with all different agricultural systems, with livestock, with cattle, with poultry, with silage inoculants, and feed preservatives and soil microbes, because we think that using natural microbes, or using nature's kind of way of keeping things healthy, is a more sustainable way and cleaner and just as effective as using chemicals and antibiotics to maintain the health and promote the health of the living things, of the living beings. And so I think that that's just kind of our mission, and that I think that's important to note.
Are you collaborating with any other researchers from other companies? For example, do you know who Paul Stamets is?
Yes. Yeah, I know who Paul Stamets is, and we're not really collaborating with them, but I think that their mission and their interests are exactly right, exactly the kind of thoughts that we put into our studies and products as well. So we try to grow. So he's growing different mushrooms and producing different mushroom extracts. And we grow different bacteria, and we produce different bacterial products. Similar things, similar approaches.
Similar approaches, but different organisms.
Different organisms.
And how are they different?
So the how are they different?
I don't mean to stump you, but
they're like different branches of evolution, but I don't think that that's necessarily the kind of answer I'm looking for. I think that the biggest difference between our approaches is that we are working only with agricultural applications. So from the very beginning, we've been working with different bacteria that could be used for food production.
Okay.
I understand that most of the work that Paul does and their company, Fungi Perfecti, is human health oriented, right? So we're thinking about how to keep humans healthy. And they're doing a great job of that. It's just we're in different areas.
Yeah, because he has been trying to work on a strain that would benefit the honeybees, but they've had problems with temperature parameters and with it even surviving in the hive. So there are challenges there.
But that was the kind of microorganism that would kill varroa mites.
Right.
Which is also a question that we tried to tackle for many years. But I think that that's a different question, right? Like you want to have something that would be like a Trojan horse that would kill the varroa mites, and that would be that would be something that would work outside of the bee. And the hive environment is pretty unique, right? So many of the microorganisms can't survive necessarily and kill the varroa inside the hive. We're looking at the bacteria that are very well adapted to living inside the gut. So that is a different kind of environment. And these bacteria are already happy inside the gut and in this anaerobic environment. So yeah, it's a much more difficult question to answer. To say how to kill the varroa mites. Yeah, we all want to do that.
Yeah, and every time you think it's about to happen, something pulls you back. But
well, I think that you were talking to some of the guests in your program actually that have more genetic approach, and I think that probably looking at the things that honeybees themselves can do, their behavior and their genetics, I think that that's a very good way to look at this question.
So another question that beekeepers, backyard or commercial, are going to want to know, does it matter if honey supers are on or off, or does it make no difference?
It doesn't make no difference. So we really, I mean, working with honeybees was very different because we eat everything, and we all of the food and all of the products that beehive produces are used in food directly, not processed, and used in cosmetics too, like wax and propolis, right? So we use only food grade ingredients, and so all of the ingredients that go into SuperDFM are safe for humans to eat as well. So there is nothing besides, none of them have any residues or chemicals. So these are bacteria that are naturally found in the foraging environment and in the hive environment. So there is no contamination. And we actually spent a couple of seasons just sending our honey for analysis to make sure that there is nothing about this bacteria that could trigger rejection or trigger any kind of test result that wouldn't be optimal for the honey and for the hive products.
When you send your honey out for analysis, are we talking about the Beltsville lab, or are we talking about somewhere else?
We used a couple of different labs. I can pull it up.
That's not, just if you had it off the top of your head, that's not that critical. And another thing that people are going to go to your website and they're going to look at the SuperDFM and the SuperDFM with the P801, and they're going to want to know, which one should I get for my backyard in Indiana or Ohio, or how would they know which of the two products, since they're both offered? I believe your SuperDFM by itself is labeled for backyard beekeepers.
Yeah, so we have different. Yeah, I think that the best approach for someone who has a backyard beekeeping will be the SuperDFM, because we have lots of different sizes. So we package it in different sizes. With P801, we originally thought that it's most important for pollinators. And when you pollinate, you have to have hundreds of colonies. And so that is packaged for in bigger sizes or in larger bags.
Okay.
And SuperDFM already has 7 different probiotic bacteria, and we know that some of these bacteria also have similar benefits against pesticides, but we just thought that P801 would be the strongest one against pesticides, and that's why we added that 8th one.
Okay. So, well, I'm glad that you explained all this, and I hope we answered everybody's questions. Is there anything else on your mind that you want to just put out there before we say goodbye?
We're working not just on research and looking at the health of the honeybees, but also on practical ways to answer some of the questions that beekeepers send us. We try to come up with ways like the microcloud to make our products a better fit for beekeepers and for beekeeping management in different styles. And so we're going to continue to try to answer more questions from beekeepers. but we also work with gardening, and one of the products that we make is for soil. And so if somebody is interested about that, also please send us our questions and we can maybe help your soil to be better as well.
The soil for gardeners, like you're saying, like for vegetable gardeners?
Yeah, vegetable gardens. I'm looking at putting it at the Pollinator Garden that we have. For the past 2 years, I've been working on that.
Do you want to talk about that for a couple of minutes? Just explain what that is, because that is kind of a companion activity for a lot of beekeepers or also gardeners, and they're growing nectar and pollen resources for their bees. So if you could touch on that, that would be great.
Yes, of course. Yeah, so we have a different blend of different bacteria which are not food grade, couldn't be consumed, but those bacteria are soil bacteria and also soil fungi. And so some of these soil bacteria and soil fungi are very important for the roots of the plants and can help the plants to absorb more nutrients and help the soil to be more biologically active. And this makes soil retain more carbon. There are some bacteria in there that are nitrogen fixing, so they fix nitrogen into the soil. And also there are some bacteria that are helping the soil to retain more water because of more organic matter in the soil.
And this information about this is also on your site?
Yeah.
Okay, so people can go to Strong Microbials.
I think that we have a soil health, we have honeybee health, animal health, and soil health. And so those are also, I think, to me and to, like you said, to many beekeepers, that could be very interesting, something that we do. Yeah,
it is something, because most beekeepers are not just into one thing, unless you're a big commercial beekeeper, but backyarders want to see the whole system. They want everything to work for their bees. Yeah. So I think that's it, Vera. I want to thank you so much for taking your time to talk to me today. And I'm sure we're going to get a lot of the wheels turning and get people looking into exactly what it is, because people have written to me all the time, hey, Fred, have you looked at DFM? Why don't you talk about it? So I thought I would just go right to the source and get the real information. So thank you so much for joining me today. And that's
Thank you, Fred.
I hope you have
It's been a pleasure speaking with you, and good luck to everybody.
Thank you so much. Same to you. Have a great night. Well, that's it for today. If you like this series, please let me know by leaving a comment down below or a thumbs up. You can also listen via Podbean, the way to be. Thanks for being here today, and I hope you'll join me for future interviews with experts. Have a great day.